Okay, so what is IndieBio? We are a sub-fund of SOS Ventures. It's a $200 million venture capital fund. We fund and build early-stage biotech companies from across the world. We view ourselves as serving all 7 billion members of humanity. We're based in San Francisco, but really our mission is to really accelerate biology for everyone. Right, that's one of the aspects. My personal mission, so I co-founded IndieBio with my other two co-founders, Arvind Gupta and Ron Shigeta, but my personal mission over the last 15 years has been finding ways of democratizing access to biotechnology through the creation of biohacker spaces, companies, and various other things. In total, with IndieBio, we have funded 55 biotech companies, and I personally have been involved in the starting of 3 biotech labs, which have led to a host of other different projects and companies as well. So I'll get us started. I want to also talk about the renaissance that's coming of biotechnology, not just therapeutics, but everything else. So biology as a technology. I also want to dispel a myth. So as you can see here, sort of, I think the resolution's a little off, but I'll explain it. A lot of people talk about the unicorns, the tech unicorns. I live in Silicon Valley. People talk about tech all the time. What's interesting is the mindset is changing in Silicon Valley. Right? And so what we're seeing now is that saying you've built the next market or the next app is no longer exciting to most of the leading venture capitalists, the ones that change the world, like the CEO, the former CEO, Eric Schmidt of Google. His excitement is actually about biotechnology and artificial intelligence. When you talk to him, and I have, we've talked to him about some of the companies that we've built. Vinod Khosla, who some of you may also recognize, Khosla Ventures, again, another billionaire investor. Peter Thiel, I've talked to all of these investors and billionaires. Their excitement is about new areas of biotechnology. And so, it's also not true that there haven't been great returns in investment. Now, actually, this is the old model. So over the last decade, from roughly 2001 to 2010, returns in biotech has actually exceeded returns across all software and IT. Again, this is not necessarily including the unicorns. So if you look at as a class, biotech actually tends to get better returns. There's fewer companies, but better returns overall. So that's kind of the picture. That was actually sourced from Bruce Booth, actually one of my favorite biotech VCs over at Atlas Ventures in the US. So what is biotech? Most people still think of biotech as an injection or a molecule that you take for a disease. Biotech is much, much more than that. This is biotech. Right? This is, some of you will recognize what this is. Probably most of you recognize it's a cell. Right? But a cell is a biological machine. Right? It is nanotech. It's molecular nanotech at its fundamental basis. This is actually from the European Molecular Biology Institute. It's a look inside of a cell. You can see all of the little organelles and functions that a cell actually has. Right? It is exquisitely complex. It's like alien technology. We're only now just getting an idea of how to engineer these systems, and when we do, they change everything. Right? The beginning of the biotechnology era in the late 1970s, early 1980s, which led to a whole boom of new biotech companies, including Genentech, which was the first company to create recombinant insulin, changed the lives of millions of diabetics across the globe who no longer had to inject pig and cow insulin taken from slaughterhouses, right? It would cause big immune reactions. I mean, people were very, very sick. Today, diabetics can live a relatively normal life. Type 1 diabetics who are born with diabetes. And of course, we have another problem now where people eat well. Today, there are more people dying of diseases of overeating, of obesity, than of starvation and famine, right? So it's a different problem that we have to deal with, and that also leads to diabetes too. So this is biology as a technology, right? This is a new era, a new renaissance for biotechnology, outside of just therapeutics. This is where we're going, right? This is where we've started to go with IndieBio. IndieBio is less than 2 years old, less than. I think we're about a year and 3/4 old. Right? We launched about a year and 3/4 old, and we have lots of great stories that I'm going to share with you. Yes, I'll try and slow down. My apologies. I'm a very fast speaker, so I'll try and slow down. Sí, y yo sé que estamos hablando en inglés, que es diferente. Apología. Discúlpame. Okay. So Biotechnología. So Biotechnology, alguna vez hablo Spanglish. Voy a tratar no hablar Spanglish. Soy de Miami y en Miami hablamos mucho Spanglish. So Biotechnology, the World Economic Forum, in its most recent meeting, obviously it's a huge meeting of all the world's leaders and pioneers in business and in politics and government, actually classed biology as a technology, as the fourth industrial revolution, right? The melding of biology and technology. It'll solve, and it's starting to solve, some of the world's most intractable problems, right? This is actually a huge shift in thinking. So actually, and this is definitely not new to Bioargentina. Bioargentina is one of the leaders in agricultural biotechnology, in the use of GMO soy and corn. I believe when we were having discussions yesterday, Argentina feeds 600 million people with the products of biotechnology. That is 10% of the world. Turns out, and I was talking to an innovator here who's doing bee pollination and innovations in bee pollination, which Argentina apparently doesn't do, that you could actually increase the yield with bee pollination by about 40 to 60%, right? So let's feed 20% of the world out of Argentina. Okay, and I need to slow it down too. Todavía estoy rápido. Perdóname. So this is a quick description. White biotechnology are industrial enzymes. This is why we no longer have what's called red tides, the algae that kill things. You need the red tide. You need the red tide. We're cutting down on the red tides. So white biotechnology is when we get stains on our clothes, when we wash our clothing, our detergents, now they're engineered. Those are engineered enzymes, genetically modified, so that they break down protein. Biofuels, unfortunately, many of them died, but in other countries, actually like Brazil, they've innovated a lot on bioethanol and other applications. This is only the beginning, not the end for biofuels. Implantable medical devices, where technology and biology merge. IBM's Watson, I don't know if you can recognize it there. Growing furniture, that's actually a chair that's made out of mushroom mycelium, right? It's agricultural waste. So we actually funded a company that turns agricultural waste, called MycoWorks, into products, like actual products that replace many things. Wood included, but also plastics and other things. That's the Impossible Burger. It looks like a real burger. It's not. It's actually fully plant-based, and it's apparently delicious. I haven't tried it. They're serving it in 2 restaurants in New York and San Francisco, but it's coming to the world soon. It's all plant-based, but the bleeding burger that tastes like blood was engineered by Pat Brown. Pat Brown is the CEO. He's actually also a professor at Stanford in molecular biology, and he engineered the burger so that it bled. And he used plant-based heme groups to do that. So I'm going to start off with examples, because I like examples that they can always challenge your mind, right? So what happens when we can program biology in a similar fashion to how we program information technology? What happens? Well, we get innovators. This is actually a team out of Princeton University. Again, we recruit globally across the world. We're just looking for interesting minds. That's the main criteria. 2 co-founders, 1 business lead, 1 science lead, and an interesting mind, an interesting idea. We funded lots of entrepreneurs across the world, including Latin America. These 2 scientists were actually microbiologists. They studied bacteria. And so what they did was they found a weird quirk of bacterial metabolism, right? It turns out that they could turn a switch in the bacteria, genetic switch, and the bacteria would go from growing, so 1 bacteria becomes 2, becomes 4, becomes X, millions of bacteria, to the bacteria doesn't grow anymore. The switch goes, and what happens is the bacteria just swells, just makes lots and lots of protein until its wall, the cell wall, expands and it pops like popcorn. Gets up to about 5 times the size of a normal bacterial cell and pops, full of protein. And so it turned out that you could actually have an entirely new protein production platform. So they came up with this idea of, what could we do with this? So they applied to IndieBio, and we helped them kind of figure out like a plan. We targeted food. We were like, what could you do? What could you revolutionize? And they're like, gelatin, right? Gelatin, for those of you that don't know, we get the scraps from animals, the skins, the bones. We put them into an acid bath. We leave them for a couple of weeks. We pull the goop out, right? The stuff that's very clear and goopy. It's actually collagen. It becomes gelatin. It gets cut up in there. And that goop, we turn that into gummy bears and sweet treats that we all eat. And it's really gross, right? It turns out it's actually very hard to control the properties of that goop too, because cow hooves and pig skins are not the same product, right? And so when you're talking to the world's largest manufacturers, they're like, we've got a problem. They turn away about 90% of the shipments of gelatin they get because it's not the quality that they need. So they thought, well, why don't we just make the protein, collagen, which becomes gelatin, and just do it repeatably. And with their platform, they did the numbers. They looked at the fundamentals, and they said, well, we can turn sugar into product much cheaper than the current gelatin market, which is very cheap, by the way. It's like dollars per pound. They can beat that, right? So they were like, okay, great. So let's make some collagen and some gelatin. So they did. They made that. And then they were like, okay, what else could we do? And they were like, let's think creatively. So with biology as a technology, you don't need to take it from an animal that existing today. So they put in the genetic sequence for mastodon. That is not an elephant. That's a mastodon. The last time humanity tasted a mastodon was about 10,000 years ago, right? So that is the genetic sequence. That's the gelatin from a prehistoric animal, right? A mastodon. And then they got crazier, and they were like, well, someone found a gummy T-Rex fossil, right? They thought it was contaminated. It turns out that the DNA was decomposed. There's no DNA, but there was collagen. And once we know the sequence of a protein, we can reverse engineer it from protein to DNA to something we can synthesize in the lab. And so what they did was they filled in the holes. There were holes in the sequence, kind of like Jurassic Park. You know, they filled it in with like ostrich, right? That's the closest thing they could find to fill in the holes. And they made T-Rex gummies. And they ate them. No one died. So this really is biology as a technology, right? So now we can do new things with biology, things that we haven't done before. And of course, these new gelatins all have different properties. Do you want it gummier, softer, harder, a slightly different property? What about medical applications? What about your cosmetics? How do you use that? Right? So these are all customizable properties of biology. And so this is actually the Memphis Meats Meatball. Again, another company that we funded at IndieBio. About 6 months ago, they made the first lab-grown pork meatball, and they ate it, right? Apparently it was very tasty. My chief scientific officer ate it too, and he said it was delicious, tasted like meat. He eats anything. I don't. I'm vegan. He eats insects, all sorts of stuff, and he loves meat. And so this was meat, grown in a lab. And that cost of that lab meatball today, well, actually 3 months ago, was $1,000 in a lab, tiny lab, probably the size of this stage. Right? So this is giving you an idea of where we're going, like today, not in 10 years. We're doing this in the lab today. When you start applying the tools of biotechnology, the long tail, huge innovation potential. Miraculex, a company that grows in lettuce, grows a protein from a plant called a miracle berry. You may not have heard of miracle berry. It's a plant that grows in West Africa. It actually grows in the jungles of West Africa. It has grown up with humanity. So fruits have a strategy for spreading their seeds. They're sweet, right? We eat the sweet fruits, and then eventually we pop it out the other end, right? And that's how they move around. That's how plants move around. So what happened was this particular species, there are several species of these plants, decided that their strategy was actually not sugar. Their strategy was protein, sweet protein, 0 calorie, right? But it grows very slowly, and there are issues around its commercialization. But the protein itself is sweet. And so the first was miracle berry. It turned sour taste sweet. Some areas of West Africa have been using this for generations. 0 calorie. Again, remember with the diabetes thing, 0 calorie. If you like sweet things, you can eat as much of this as you like, 0 calories. But the challenge is making it. And so it turns out that he can make it in lettuce, recombinant lettuce. Simple, right? Solve the problem. Not so simple, a little harder than that, but you can make it, right? And what's unique about this protein is that it is a protein, big protein. It actually fits onto the taste receptors of your tongue. And what it does is it's a molecular machine that tricks your tongue into thinking it's sweet. And it only seems to work in upper primates. And so that basically means somehow we, primates, upper primates, have evolved with this plant over, millennia, right? Way too long. This is Clara Foods. They actually brew egg whites, la clara de los huevos. And so Clara, one of the co-founders is Mexican, Arturo, and he actually had this idea of taking the genes from chickens, putting them into yeast, and brewing them like you brew beer, right? So he came to us and he was like, we're gonna do this, and I was like, that's so crazy. I looked at the science, and my co-founders looked at the science, and we're like, it should work. So we did it, and they raised funding. They raised 1.75 million when they first graduated IndieBio within 3 months, right? And then most recently they raised this is not public, but they raised a large Series A. I'll leave it at that, right? And so it's backed by some of the biggest names in Silicon Valley. And of course, agricultural biotech, Syngenta, Monsanto, and there are many other small innovators as well. Lots of opportunities in that space, especially with CRISPR technology. So, we'll move forward. So we found something else out. When we were tinkering in the dark matter, right, of that space, más despacio, okay, más despacio, encontramos algo nuevo, ¿verdad? When we were looking at the dark matter of fermentation technology, turns out that you can actually drop the cost of fermenting almost anything, right? We were talking about fermenting like beer, right? From thousands of dollars per pound to dollars per pound within 15 years. 15 años, right? 15 years, roughly. And that was, we were looking for Moore's Law, and we think we found it for molecular biology. It's the improvement of the strains, the machines, the factories. With time, we can improve them, make them more efficient. So that basically translates to the cost per pound of sugar is about 15 cents to 30 cents US dollars, right? Per pound. That's very cheap. And that, how efficiently you turn those carbons into something else, into X, whether it's egg whites, whether it's biomaterials, whether it's other things, it can drop to dollars per pound. So what we realized is you could profitably feed the bottom billion, the people who earn the least money in the world, and profitably feed them, right? With the foods that we are very used to and love. Meat, egg whites, gelatin, non-animal products too. We can do that like today. And we're trying to collapse that curve from 15 years to maybe 5 years, maybe 3 years. We think we can do that. We, as in we humanity. We've seen it in the lab, and we've actually seen when you accelerate biology, we can do it within 3 to 5 years, no doubt, right? So biomaterials, new area, right? New area. That thread looks a little bit like silk. It's not. It's spider silk. So it's spider silk. So the founder, Bolt Threads, the CEO of that company, he doesn't milk spiders. He doesn't find lots of little spiders and milk them. He puts the genes into yeast and then makes spider silk. What's cool about spider silk? It's 10 times stronger than steel per weight. It's a material we don't have on this planet, right? That we can't use. Humanity has not been able to use, but biology has solved the problem for us already. So why not use what biology has done? And the best part is there are thousands of types of spider silk. We're only just scratching the surface of what we can build. But biology solves many of the problems we don't know we have yet, right? It's solved it millions of years ago, right? And now it's ready for us to figure out how to use it. And the best part is we can go beyond evolution. What happens if you change? What about if you want to make it conductive? Could we do that? Maybe, right? Then what, right? Then we have material that's essentially bulletproof and can conduct electricity or something like that, right? But this is real. So this was made. They're making garments now, and they're producing it at scale. This is MycoWorks again. This is actually their product. Instead of wood, that's agricultural waste treated with mushrooms, and they use the mycelium. That's the root structure of the mushrooms to make woods, alternatives. This is what it looks like. It's not very pretty. Apologies. But it works, and it's a really strong material. This is a biocement. It's actually still a research project at the university in the Netherlands. The biocement, when it breaks, it has spores, bacterial spores inside, that reawaken and calcify the concrete, right? So it's self-healing concrete, right? And then brewing things like nylon, right? So we funded a company, Zymochem, that actually does that as well. So you can see this is happening across anything that biology can make, we can make too. And this is one of my favorites, computation. So computation is a really interesting area for biology because it turns out that we do it really well. We have more power in here than any supercomputer cluster in the world. We have more power in one human brain than the entire world has distributed across supercomputers, personal computers, everything in one brain. And this runs on watts, right? And so how do we harness this power? So one company we funded, Kanaku, he was a Nigerian entrepreneur. Nigerian, trained in Switzerland, lived in London, was doing research work in London. He was trained in human-machine interface, right? Connecting the body to machinery. He decided the body's a very messy place. Can't I just pull the neurons out and put them on a chip, right? He was a PhD. He was lost in the system, not going to be a faculty member. He was a researcher. And so what he did was he basically found a way to connect neurons. He took skin cells, reprogram them, turn them into neurons, as Gabriella was saying she was doing, right? Turn them into neurons. Stem cell technology, we know how to do that well now. And then put human neurons into a chip and made it do computational things, right? Not very exciting computational, like 1960s transistors, computational things, but still computational things. The first applications for his technology is actually threat detectors. So neurons can learn what smells are, and so can smell things like TNT, gas leaks, other things. That's the first application for Kanaku. We're also looking at the brain and understanding like how the mind works to push the frontiers of artificial intelligence, right? The mind is a unique system. It is not a machine in the same way that we operate. So we need to understand it. We actually don't understand intelligence or even how one neuron works, right? So when you hear about these neuromorphic chips that everyone talks about, that IBM are making, those are not neuromorphic. I mean, they might look a little like a neuron. They don't function like a neuron because we don't know how a neuron functions, right? So we're trying to understand the electrical signals within the brain at the single neuron level and at the higher neuron level. And actually, one of my favorites of the recent batch that we funded, so again, we funded 55 biotechs. This is our most recent batch of 13 companies, is catalog technologies. You can't see this very well, but what you can see there is a data center, right? That's what we can store today, right? So we're talking about, this is on a logarithmic scale of data. So this is hard disk, flash, we're talking about a logarithmic scale of like 10 to the 10, right? Data is rapidly increasing. I think it's by 2040. Every year we will create more data in terms of bytes than there are stars in the sky, in the night sky, right? Basically, we can't store it. Intel did a calculation. We run out of silicon to meet our memory needs by 2040. Physical silicon. We run out. We're done. Can't store any more data, right? So where do we go? We funded a company out of MIT. One PhD student, one postdoc. And what they're doing is they are storing data in DNA. DNA is the most data-dense storage medium we know of in the universe. It actually stores at 10 to the 16. So you could basically store most of the world's data centers, these huge complexes over many hectares, many acres, in a little tube. You can't see it, but it's an Eppendorf tube, right? And so you could store all the world's data in one refrigerator in someone's lab. Done, right? Easy. Until we start using more and more data, right? They actually have technology that is not based on synthesis. For those of you that are scientists, you know that the cost of synthesis of DNA is very expensive. It's 3 to 5 cents per base pair. Essentially per bit, it's 3 to 5 cents. It calculates out to like over $1 trillion to make what's called 1 petabyte, right? But with Microsoft calculated out to store 1 billion terabytes, it's about 1 gram in DNA. 1 gram, right? And so what they've done is they're not synthesizing the DNA. They're writing right on top of DNA. They write on top of it. So you just PCR a bunch of DNA and you just write on top of it. Super cheap, almost free to PCR the DNA, right? So Hopefully it's kind of interesting, a little different, right, to the usual therapeutics. Let's talk about curing disease, which is very important. But biology is a technology that will disrupt multiple verticals and industries, pretty much every industry we have on this planet today. And it'll also take out things like environmental damage. It'll make things affordable. It is very scalable technology. And medicine, and I won't go into too much detail on medicine because many of you are very familiar about different aspects of medicine. But this is Orfidia, single drop diagnostic, single drop of blood diagnostic. Some of you may have heard of Theranos. This is actually Theranos technology that works, not Theranos. There is a lot of work going on in organogenesis. So organogenesis, that is the creation of new organs. Why do we die? If we cured all cancer on this planet, we'd still die. We'd get, I think, an extra 3 years. 3 years, right? So what kills us? Our organs fail, right? Our heart fails, our liver fails, our kidneys fail. We have to rebuild them. And we've made incredible progress so far in rebuilding organs. Still not fully functional, but there's incredible promise that one day, hopefully not too long, the estimates when you talk to experts is between 5 and 20 years of functional organs. At first, we're going to start very simple and move up the scale. And then implantable devices as well. Longevity is a brand new area which is evolving. There are actually some promising preclinical candidates for new therapeutics that can extend human lifespan. Some of you may be familiar with two molecules, rapamycin and metformin, old molecules, but that seem to have an effect and increased lifespan about 15% in multiple different species when you take it. And then of course CRISPR, which has the potential to transform almost everything in human medicine, in agriculture, and in many other areas as well. So this is actually a pathway. I won't go into too much detail about this, but what's fueling this renaissance? It's actually frustration, right? It's basically, the post-apocalypse, where when these scientists graduate, they can't get faculty jobs, they cannot become profesores or investigators, they're stuck. Many of them go off and do something else, but their first love is for science. And so where do they go? Well, you can't see it, again, you can't see it very well on the slide, but about 50% of scientists in the US, and I would assume here too, in Argentina, want to go on and become faculty at the university. They want to be faculty. Only 1 out of 10% will ever be faculty. 1 out of 10. So where do the rest go? A bunch of different places, not faculty, right? So what's led to that is if you want to do your science, you have to find another way of funding that. And as has worked, capitalism is the least perfect system we have, but it also works. And so if you have a commercially viable idea, you can try and find a way to make your science sustainable by funding it, by getting it funded, and by getting customers and by making revenue. And so that's what started to happen. So we've seen postdocs leaving the post-apocalypse. We've also seen low-cost biotech. Projects like the mini PCR have enabled low-cost biotech and the drop in costs. So tech has actually, Moore's law has enabled us to do science cheaper and cheaper and cheaper, and that enabled myself and many others to create biotech labs, biohacker labs. Many of you may not be familiar with the term biohacker or DIY bio, do-it-yourself biology. It's spread globally. Back in 2009, it was only 1 or 2 labs, one in Silicon Valley and one opened up in New York. Today, it's dozens of labs globally, including one here, Bioargentina, right here. So it's actually, they're based in Buenos Aires, but they're there. And the projects that we're doing in these types of labs, so this is BioCurious and Counterculture Labs, both based in Silicon Valley, making real vegan cheese, brewing cheese. The Open Insulin Project, I actually helped co-found that with Anthony DeFranco, and it's to make insulin free. So anyone can brew it, you just have to have a culture, right? Today in the US, and I don't know what it is here in Argentina, but in the US, a diabetic has to pay $500 a month if they don't have insurance. So why not brew it? It should be the cost of, a little over the cost of sugar water, right? Why $500 a month, right? So we want to enable people to have access to the tools. And a big problem has happened that, if you are in a war zone or you have problems with logistics in your country and you're diabetic, you'll die. Right? And that has happened many, many times. So many millions of people still die because they're diabetic. So why don't we help them with the tools of science that we have? These are the the biotech labs all across the world. It's just a snapshot. There are dozens of them all over the place. In Paris, in London, in LA, in Silicon Valley, in Buenos Aires, in Australia. They're everywhere. All across Latin America. There's actually a Latin American biotech network for biohacker spaces too. And I wanted to talk about, can biology be exponential? So a few of us have already seen this slide. This is sequencing costs. This is just to get to the point that biology can move faster than Moore's law, right? This is Moore's law. This is sequencing costs, right? So if you project it out past 2015, 2016, eventually within a couple of years, it'll be cheaper to flush the toilet. It'll be cheaper than flushing the toilet than sequencing your genome. Right? Maybe have an analysis as it goes all the way through. Every day, every flush, one analysis per flush. So biology can beat Moore's law. It's not just sequencing, it's synthesis is dropping, not as fast. It's a problem, by the way, that we have to solve. Synthesis is very expensive. Cells in our bodies do it for nearly free every day. Millions and billions of cells do it. We need to get better at it. Digital PCR, CRISPR-Cas9. This is an amino lab. This is a desktop bio lab for everyone. And so this allows you to engineer bacteria at home. This is Julie. She was the CEO. She was a designer that came out of MIT's Media Lab. And she applied to us and she was like, hey, I want to make it accessible to everyone. I want everyone to be able to do genetic engineering at home, right? A few people were like, at home? No, not at home. But that's what we support. So we were hoping to democratize the tools. This is Joey Ito. He's the director for the MIT Media Lab. He basically says that biology is moving about 5 to 6 times faster than Moore's law. Right? The same Moore's law that turned big clunky computers that were the size of nearly half this room into supercomputers that we had in our pockets today. Right? So faster. So 5 or 6 times faster. So what does that mean in 5 years? It means how we use biotechnology will be almost impossible to predict in 5 years' time, in 10 years' time. It will be transformed. And of course, the leading innovators today, the big global giants are noticing. So AbbVie bought Stemcentrix for $10.2 billion. That was the cancer therapeutics. That was actually the largest venture capital deal in history. A few friends of mine that work at Founders Fund made a lot of money on that deal. There's also Bolt Threads, which has raised incredible amounts of money, about $60 million. Google tried to buy Impossible Foods, that burger that I outlined. They tried to buy them for $300 million. Pat Brown, the CEO, said no. He says the market potential is way higher than that. When you're serving global markets, 7 billion people, 300 million, just to toss in the ocean. And this is my favorite quote from Hunter S. Thompson. So I like the weird stuff, right? So this guy's stretching out. He's in one of these huge mega cities in Beijing, in China, and there's no honest way to explain the edge because the only people who know are the ones who have gone over, right? So I encourage you to go over the edge, like to find out. This ecosystem in Argentina, money's not the issue, right? It's mindset. You have enough money in the ecosystem here, right? You have enough wealthy investors here who need to, by the way, cut checks to support the next generation of entrepreneurs. That's necessary. Tienes que hacerlo. But also you need to give them your advice too, right? They need help. So those first-time founders, fund them, help them. Yes, it's risky. Most will fail. You have to. That's how you make huge returns. And that's also how you encourage the growth of an ecosystem. And so we're already seeing that here in Argentina. It's here. The science is here. The scientists are here. The money is here. Just a mindset shift. Click. And the Nakama is doing a great job. I know there's a few other ventures, company builders being built out. And I think that's going to be a great thing for the Argentinian economy. There's no reason why Argentina can't be a leader across Latin America and across the globe. So thank you very much. Muchas gracias a todo el mundo.